Thank you very much to the organisers for the opportunity to present precisionLife's work in myalgic encephalomyelitis today. I'm going to start with a warning. This is not my work. The really smart people whose work this is, Shirley Das and Steve Gardner, are in Salt Lake City this week for reasons that will become clear during the presentation. So I do apologize that you will get my version of the story. So my agenda today is to give you a quick introduction to precisionLife's approach to analyzing patient data for chronic diseases, and then give you some insight into our timeline and how we got interested in ME, then talk a little bit about what's next, and certainly spend some time on acknowledging the generosity of people in this community who've been incredibly helpful in helping us relate our findings to the world. So precisionLife, we set out to focus on complex chronic disease because these are the diseases that account for 80% of all healthcare costs, which means that's what most of us are going to be impacted by at some point in our life. These are the diseases that shorten all of our lives. And to the point that the last speaker made, if there's any bright light here, there is an awful lot more patient data available than there would have been a few years ago. And we are very fortunate to be able to work with various organizations to access patient data. Some of that is relatively public, accessible to anyone through the right approvals. UK Biobank, dbGaP collections in the US. Also working with patient charities. You'll see Action for ME on the slide, and I'm very fortunate to be working with the Decode ME study group. Our basic hypothesis is that the established methods for analyzing genomic data and other omic data have a particular assumption that we do not believe is appropriate for complex chronic disease. So genomics has had fantastic impact in the field of rare disease and oncology because those diseases are monogenic. So you can look for a single feature across a whole population, find it in a gene, and you're going to get real insight into what's driving the disease biology. But as we've heard today, ME isn't like that. Our patients present with multiple different phenotypes, and this is common to chronic diseases. Patients who share the same diagnosis can have one or more underlying disease drivers that give them that clinical presentation. So we are looking to find combinations of markers that are statistically significant for subgroups of the patient population, and through that achieve a hypothesis-free mechanistic stratification of the patient population. So here's a picture of what that looks like to us. So all of the patients whose data contributed to this analysis have the same diagnosis, Alzheimer's. And if I was to analyze that data set from UK Biobank using GWAS and very well-established tools, I'd find 10 SNPs, as was previously described, 10 genetic variants, all in the ApoE locus. And that observation has really been the foundation of the pharmaceutical industry, spending billions of dollars pursuing amyloid for the treatment of Alzheimer's, with very little success. With our approach, by looking for combinations of features, data features, in this case SNPs, in specific patient subgroups, and then overlaying the patients who have common SNPs, we're able to cluster up the view of the patient. So these colours here represent patients whose disease is being driven by common biology. And when you dig into the genes that are in each of those colours, yes, you do find the signal associated with the amyloid hypothesis, so lipoprotein metabolism is the biggest driver of disease, but only in a rather smaller percentage of the population than you might imagine. But we also find signal in post-viral neuroinflammation. Here's the clue, guys. Similar signals that we find in severe COVID, and also autophagy, copper iron homeostasis, brown fat metabolism, and a serotonergic neurotransmitter mechanism. And if you go into the literature, you will find evidence that there is a hypothesis around these mechanisms. The results we will put in our repository, our proprietary repository. We imaginatively call it our DiseaseBank. And the insights that we're able to drive, we use to expand the knowledge with partners around known targets. So can we tell them about prevalence? Can we talk about the directionality, the causality, the case for causality around that target? And also identify novel target biology. The patterns of SNPs describe the mechanism of the genetic mechanism behind the disease, and we believe that this is potentially a mechanistic patient stratification biomarker that can be used in clinical research and may have application in diagnostics. So this is an incredibly scalable approach. So we've undertaken over 50 disease studies in a range of chronic conditions. You'll see in the bottom left-hand corner the kind of diseases that we've been looking at that are perhaps relevant to the audience today. And that view allows us to spot patterns of disease biology across different diseases. So the matrix on the right shows 6 different CNS indications. So from the top, ALS, Alzheimer's, bipolar disorder, frontal temporal dementia, Parkinson's disease, and schizophrenia. And you can see here how we're seeing the same genes active in multiple diseases. And then the images below are a view of a particular novel drug target, a GPCR, that again, we can look at being implicated. We find signal that they're being implicated in a portion of the patient population in bipolar disorder, Alzheimer's, schizophrenia, and migraine. The other thing that we can do, because we're finding known target biology where we have drugs, is we can start to go hunting for where those targets we find in one disease may be active in another. So here we've got a target, there's a known drug, it's being developed in a particular indication. We can look in our DiseaseBank across multiple diseases and identify a different indication where that target is implicated. We can look at whether the prevalence of that mechanism is interesting, whether it represents a patient group whose clinical needs are unmet, and if that looks like an attractive hypothesis, we can go and look at various commercial considerations for making the case, perhaps with a biopharma company whose drug is marketed to expand a label or to add additional indications to clinical trials. So that's the broad capability that we have in precisionLife. And I just want to talk now as to kind of how we came to the ME/CFS challenge. There is a QR code which will take you to a page on our website. You'll find publications and posters and news and updates of what we're doing in the field there. So that might be of interest to you. So it all begins back in the pandemic when UK Biobank uploaded its first data set for severe COVID patients. And precisionLife, like many companies and academic groups around the world, jumped on this data. We agreed as a business that anything we found we'd just put in the public domain and see if we could help. And we were really trying to understand the host response to the virus and how certain people were having very severe forms. And we put a preprint up in June 2020. We implicated 68 genes that were interesting because they linked to the observed clinical symptomology. So in addition to the genes that you would expect in a viral infection, like cytokine cascades, we found signal for endothelial dysfunction, lipid dysregulation, neuroinflammatory markers. And these, the genes that were in each of those mechanisms of action were highly suggestive of some of the conditions that people were reporting on the ward. We then did our repurposing piece. We published a list of potential repurposing drug candidates. Some of those went into clinical trials. We were the first to suggest dutasteride. Dutasteride read out in a double-blind RCT and was shown to reduce the severity of long COVID infections and reduce the recovery time by half. So we thought that was a good piece of work, and we're very keen to replicate that to the statistician's point. All very good and well doing that once. Does it replicate in other data sets? We were approved for have access to a particular data set in October 2020, and we waited and we waited and we didn't get access to it for reasons we won't dwell on here. But at precisionLife, we are many things, but we are not patient. We're about precision medicine, not patient medicine. And so, we were thinking about how we could move this on and thinking about other post-viral chronic conditions and where we could get hold of data. And so at that point, we started, well, we had two things going in parallel. We went back to UK Biobank. There are some studies in UK Biobank. They're very well analysed studies, and we were able to analyse those. I'll talk about those in a bit more detail. We were also working with Genotypic to analyse their GOLD study, which was their long COVID study, and we published that in October last year. Since then, we've won an Innovate UK grant for a program we call LACOME, Long COVID ME. This working with Action ME and the University of Edinburgh, specifically Professor Chris Ponting, who's the PI on the Decode ME data set. And we have now a replication data set, a very high quality replication data set that we are working on. So that's very promising. And we have announced in February this year a partnership with the Metrodora Institute to start planning to move this into the clinic. So just briefly reflecting on the work that we did with UK Biobank, there were two studies that we were able to identify. One is an experience of pain questionnaire, where individuals have shared their data. They have self-reported ME/CFS diagnoses. And there was a second set, which is called the CFS, self-reported chronic fatigue syndrome, where there was a verbal interview done at an assessment centre, and we were able to look at that data set, exclude individuals who were in the experience of pain data set to basically create two separate populations. We then built out the controls, obviously genetic, sex and ancestry matched, and then use the pain questionnaire as our primary analysis and rely on the verbal interview as a secondary disjoint data set. So the analytical approach runs like this. I'm going to talk about our pain questionnaire result. So we build out the data set and start by an approach called combinatorial analysis. So we're looking for combinations of features that are statistically significant for a group of patients, and then we cluster them together, the patients together, or the SNPs together, where they co-occur, to build out these clusters that you can see in that circle. And the clusters are made up of, there's 84 what we would call combinatorial risk signatures. So those are the primary groups of signal that we find. There are 25 critical SNPs, and a critical SNP is one that we see connected to multiple points within the clusters that are at the heart of conserved evolutionary biochemistry that these clusters seem to find. Those prioritize 14 genes, and the case coverage of the study was 91% of the patient population, which is a good start. Just to talk a little bit more about those clusters. So the combinatorial risk signatures, those are combinations of 3, 4, 5 SNPs. We very rarely see any biological signal in pairs, so it's a higher order set of clusters. They have persuasive p-values, and the odds ratio, the sort of predictive value of each of those clusters is well over 3, and that compares very favorably with the kind of results you can get from GWAS. None of those SNPs are in LD, and 95% of them are in non-coding regions of the gene. So just something to think of if you're building out collections. I would tend to advise you not to think about exome-based sequencing. Ideally, something that's representative of the wider genome is possibly more useful long-term. And then we move into biological annotation, so the SNPs to genes, mapping the genes to the mode of action, mechanism of action category. And we completed all that. We thought, gosh, that's good. And then we thought, well, is it? Because we don't know anything about ME. What's really should be obvious to you listening to me at this point is we are data experts. We're not disease experts. So at this point, it was really time to phone a critical friend or two. And I've got to thank Chris Ponting for being kindly encouraging and professionally skeptical in talking to us. And with his encouragement, we decided to publish our findings, very much echoing the words of Professor Dame Janet Thornton, who you will have just seen the Thornton building as you came onto site this morning. Janet was Steve Gardner's PhD supervisor and is an absolute believer in publish or be damned. You either publish it and get proved wrong, or you shut up. So we published. The mechanistic stratification that we found, this is hypothesis-free, but it allows you to do something really, really interesting. Our findings are linked to patients. So that green group that you can see at the top of the slide, that central SNP that seems to be linked to other pieces of biology, that SNP is mapped to a gene called ACAP1. This cluster represents about 27% of the case population. And the role of ACAP1 is it regulates mitochondrial energy function by replenishment of ATP through the AMP kinase enzyme. And so if you had a defect in that gene, you might suppose that those would be the patients who would present at clinic complaining of post-exertional malaise. And so we're able to look at the annotation for the patients or the patient in the patient record, and indeed those are the patients complaining of post-exertional malaise. So that's really interesting. A hypothesis-free link from a genetic observation to something that's potentially explanatory of clinical phenotype. And that's similar thing going on in the blue cluster over here. that central SNP is mapped to a gene called, well, it's not really got a very good name, but anyway, it's involved in neurotransmitter transporter. It's an enzyme called GAP3, or receptor called GAP3. If you've got a defect in your neurotransmitter signaling biology, you might expect people with a defect in that gene to present in clinic concerned about cognitive impairment, struggling with mood disorders, and you'd certainly expect to see increased neurotransmitter levels in their records, and that is what we see. So this is very encouraging, and you know, please read the paper. It's all there for further digestion. One other piece just down here, this pentagram little cluster, that's clock. Obviously a master regulator of circadian rhythm. Interestingly there we saw significant co-association in that group, also having a fibromyalgia diagnosis. So moving on to the long COVID study with Sano. So Sanogenetics collected a long COVID data set. We were able to look at building two study cohorts from that data. One is a severe COVID cohort, so those were the people reporting the most acute symptoms as a result of their long COVID, and a fatigue-dominant cohort, so the individuals who were challenged by fatigue. In the overall, we found 73 genes highly associated with the two phenotypes. We found evidence for 14 of them being differentially expressed in a prior transcriptomic analysis. 9 genes had prior associations in the acute COVID study that we'd done, and 9 had been identified previously in our ME-CFS study. Interestingly, there was a higher overlap of the ME-CFS genes in the fatigue-dominant cohort, including the ACAP1, insulin receptor, and clock cohorts. So what's next for us? Well, we're interested to see whether we can prove out the predictive value, not just for the individual mechanisms, but the whole model. It's not a true diagnostic. This is something that might support clinicians triaging people presenting in clinic with symptoms that may or may not be ME-CFS. And really it's describing a lifetime disease risk of the disease, but also giving insights into potential mechanism. And so if you do something as simple as count how many times those combinatorial risk signatures, those 3, 4, and 5 SNPs appear in cases versus controls, you see a very clear difference in distribution. In fact, the odds ratio or predictive value of somebody with a ME-CFS diagnosis having multiple of these factors is 8.9. For comparison, the BRCA gene has an odds ratio of between 5 and 7.5 in predicting breast cancer. So that really is quite encouraging. Because we're working with UK Biobank data, this is all genotype data where the patient's genotype was measured using the Thermo Fisher UK Biobank Axiom array. That's a standard catalogue item, £30. Sorry, I'll rush through. We've now got a collaboration with Metrodora. Just very quickly, we are currently analysing the DECODE-ME data to build a risk score that includes not only the risk genes that we've identified, but also looking at protective effects. In parallel at Metrodora, we're doing the work of recruiting patients, genotyping them, so that we can look at whether those risk signatures coming out of the DECODE study has clinical utility for confirming whether a patient is at high risk, and clinicians can use that. And we're then planning a second study early next year where we are going to sub-select from those initial patient groups Where there are mechanisms that are highly prevalent, where we have safe and well-tolerated drugs, these will likely be generics in the first instance. We will have clinician investigator-led trials operating at the Metrodora Institute, where we will dose patients where there is a phenotype where we believe we can get a fast readout in order to prove that we can select patients around a target that has disease-modifying potential, so that we can advance some of those medicines into a full clinical trial. Why are we doing that? We think we desperately need tools that allow us to stratify patients to support the wider clinical trial efforts of this community. We certainly need treatments. If we have treatments, then we can have a reimbursement case around these tests. And because we want to bring the pharma industry into this situation, into this indication, if we have clinical validation in people with ME that we have target biology that is disease modifying, and also validation that we can select individuals who will benefit from those drugs in those mechanisms, we have massively de-risked biopharma's investment in our indication. And I think that would be good for everyone. I'm going to not, we're planning to get this read out by the end of the year. I do want to say we can't do anything without patients who often are inconvenienced by participating in these trials. So thank you to them. Certainly to our experts and those who were friendly critics, particularly people like Simon McGrath, who's a scientist living with ME, who wrote the most wonderful, accessible description of our study. So thank you to Simon and Ovid, who connected us to Vicky Whitmore and invited us to participate in the Future of ME roadmap for research. And most of all, my fantastic colleagues at precisionLife whose work this is. Thank you very much.